Hi class, it's Jamie Batts, and we are going to cover the next part of our chapter about blood. So we just finished talking about how blood cells are formed. We're going to quickly review some of the lab type things that you can do to analyze blood, some of the tests, and then we're going to spend the rest of this video talking about red blood cells, what they are, how they're made, etc. So switcheroo here, and there we go. This is section 17.4 if you're following along from the Martini visual textbook. So hematology is the study of our blood and blood forming, as well as blood disorders. They're all related in this study of hematology. So our blood, because it has such an important role in our body with transporting and delivery of waste products, this is one way we can really analyze and see what's going on in our body. If there's some type of clinical case where, someone's presenting any issue that can't be easily diagnosed topically or however, the blood is going to give us a little peek into the rest of the body. Blood and urine, any body fluid really can help give you this. But because our blood has such a huge role in our immune system, it gives us that much more information than any other body fluid that we have. So let's look at why we would want to perform blood tests. So first, determining blood type, and we will talk about how to determine blood type in the next video. Will also can be used to evaluate just the different numbers of red blood cells or white blood cells, as well as platelets. So it's helpful in diagnosing in that respect. Any abnormal values can indicate disease, and depending on which value, or is it up or is it down, or is it this cell up or that cell down, that's going to give us sorry, my nose is crazy itchy right now. That can even clue us in a little more as to exactly what the pathology is. Something like a CBC, or complete blood cell count, that's going to give you the specific number of red blood cells and white blood cells, erythrocyte indices like hemoglobin content, etc. A hematocrit, which is your ratio of plasma to formed elements, or a differential count in general, just the different types of and numbers of each of the white blood cells. So all very helpful. In a complete blood, this is an example of a complete blood count. So you would get the hematocrit, which is your percent your percent red blood cells, your hemoglobin, which is just the overall concentration of hemoglobin in your blood. Typically, if your hematocrit's down, your hemoglobin's going to be down also. So they look for congruency in those two numbers. And then they're also going to look at just the different types of white blood cells and their overall concentrations, platelet counts, etc. Overall red blood cell tests, there's several common tests for red blood cells. The number, the size, the shape, right? They can easily diagnose by looking under the microscope and doing a little bit of chemical analysis. They can produce, they can give some indices of disorders that can't just be diagnosed through very obvious symptoms, like internal bleeding, for example. So this is the different types of tests that can be done and the overall basic diagnosis that can come about because of elevated or depressed levels of those numbers. Again, do you have to know specific numbers? No, but in a case study or an essay question, I might give you a value and tell you, blah, 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 this is the number which is high or which is low. And then you would have to kind of understand from that and interpret that from there. So you can pause the video here. I'm going to keep lecturing, but you can pause the video here, go back and answer these 3 questions about this section. And I'm going to keep talking about red blood cells now. So red blood cells, erythrocytes, RBCs, all the same thing, about 1/3 of all the cells in the entire body. A huge concentration of these guys. Single drop of whole blood has millions, around 260 million red blood cells. So the average adult has trillions of red blood cells inside of their body. Kind of crazy. So the red blood cell count is just a standard blood test. You're just literally looking at the slide and doing a representative sample. This reports the number of RBCs per microliter. Adults, males and females. Males tend to have higher values for this because they have larger bodies. Larger bodies require more blood, so makes sense. there is a stained blood smear, and here, and if you're in lab, you've already looked at this already, right? These guys are our red blood cells, and you can see some platelets in there, those little fragments. And so some overall characteristics, they're a biconcave disc, so they're caved in on the top and on the bottom, so they're kind of flattened in the middle, almost like a donut, but without the hole in the middle. Average diameter is about the same, so they're very uniform, and they're all identical, right? They lack nuclei, so they're anucleate, and they lack most other organelles. So they don't have ribosomes, they can't repair, they can't divide. Once they're damaged, they're done, right? So the lifespan on them is less than 120 days. So roughly every 4 months or so, those red blood cells go over to the spleen and they're recycled, removed, and some of the parts are used to rebuild blood in the red bone marrow. So they are recycled. And again, that's because they can't undergo mitosis like the rest of the cells in our body, or like most of them, really. So there you see the overall dimensions. You don't have to know numbers. We'll move on. Some functional aspects. So in other words, why are they biconcave, right? This is for 2 reasons. It helps increase the surface area. If the cells have a higher surface area, they're going to basically have more doors and windows to get into and out of the cell. So this is going to allow the transport of materials into the cell and out of the cell. Highly efficient for transport, which is one of the main functions of blood, right? So the total surface area is huge in these red blood cells compared to their inner volume. And because they are biconcave and they're all uniform, they can form nice little stacks or rouleaux. These help kind of transport them through very small vessels like the capillaries, which are very, very narrow, only 1 cell thick. And so these red blood cells, you can almost imagine them like lining up and just passing through in little like stacks, like poker chips, through the capillaries there. They're also flexible, so they can kind of move and twist as they need to get through smaller capillaries. So that's just a summary of the functional aspect. Here you can see those rouleaux, those nice little stacks in there. This is a cross section, so that you can kind of imagine the capillaries like flowing out at you or into the into that picture there. So it's a section of it. Let's talk about hemoglobin. The primary function of hemoglobin is to transport our respiratory gases. So it's a major molecule, and this is random, I had a hemoglobin molecule on my desk, but I can't find it right now. Oh well. So it's I make little models of things because I'm super nerd like that. It's the major molecule that's responsible for transporting carbon dioxide and oxygen. So it accounts for more than 95% of everything inside of the red blood cell. So basically, when these red blood cells are formed through that process of erythropoiesis, everything's taken out of the cell except hemoglobin, pretty much. Hemoglobin is stuffed in there, jam-packed in there. So again, males have a slightly higher hemoglobin count than females. They have more red blood cells, so that makes sense. And because males tend to have larger muscle capacity, they need more oxygen for their muscles. So there you see it. This is what my model looked like. I actually had a pipe cleaner that was all like combobulated around each other, kind of like this picture. And I had some little beads on there to represent the heme. So the heme is our functional binding unit that contains iron. And we're going to start talking about that right now, actually. So hemoglobin is a protein, obviously, and it's basically made of 4 polypeptide chains that are stuck together. 2 alpha chains, 2 beta chains. Each chain has its own heme molecule on it. Heme is actually a pigment molecule. So there you see the light blue and the dark blue, and then 1 heme for each chain. So each heme has an iron ion on it. So the iron interacts with the molecule of oxygen. It forms oxyhemoglobin. It gives the oxygenated blood its bright red color. So the dark blood, and you may notice this if you've been around blood a lot, the dark blood is typically low oxygen. If you've ever donated blood, they're tapping your antebrachial veins right there. It's typically like a very dark, almost like a brick red color. It's because it's the low oxygen blood. It's in the vein, right? Oxyhemoglobin is very more concentrated in your arteries, which are bringing fresh blood to your tissues. So the binding of iron and oxygen is easily reversible. So think of it almost like a, I forget the name of the game, but it had those little mitts that had Velcro on them, and then they had those little like tennis balls that would stick on and stick off. It's just like that. The heme is the little mitt, right? That little Velcro mitt. I think it was called Scratch or Sketch. I don't know. Anyways, so you have that little mitt, and then you have the ball that just comes on and off. That is hemoglobin and oxygen. Pop on, pop off, pop on, pop off, as needed. So hemoglobin that's not bound to oxygen is called deoxyhemoglobin. So you have oxyhemoglobin and deoxyhemoglobin. That is giving the deoxygenated blood its darker color, like I mentioned earlier. So there's the heme, there's the iron, and iron is just there to increase that scatchability, right? That attachment of oxygen to the heme molecule. So hemoglobin, again, contains millions of these hemoglobin molecules. Red blood cells, I should say, contains millions of these hemoglobin molecules. So if we have a red blood cell that has millions of hemoglobin in it, and we have millions of red blood cells, well, I mean, that's like billions and billions and trillions, really, because we have trillions of cells. That is like limitless, almost, oxygen in our body. Each hemoglobin contains 4 heme units, so each red blood cell can carry more than 1 billion molecules of oxygen. Whoa. That's in one red blood cell. So about 98% of oxygen carried in the blood is bound to hemoglobin. The rest of it is dissolved in the plasma. So our blood can dissolve in the plasma if for some reason something's wrong with your red blood cells or something's wrong with your hemoglobin. You still can get oxygen to your body, just not as efficient. So you can pause the video here, go back, review, answer these 3 questions. We're going to keep marching on and talk specifically about red blood cell formation and recycling. So, basic overview of red blood cells, about 1% of these red blood cells are replaced every day. So remember, we said there's a life cycle of 120 days, about every 4 months. It's not like every 4 months, boom, you have new blood. It's a rolling cycle. So at any given day, about 1% of your red blood cells are being broken down and 1% are being produced, right? So it's a rolling cycle. About 3 million new red blood cells enter circulation every second. Crazy. This rapid replacement is because of that super short life cycle of only 120 days. How are they broken down? Well, they're basically engulfed, eaten up by macrophages, which are large cells. They're like Pac-Man, and they are located in the spleen and the liver and the bone marrow. And so as the blood circulates there, those macrophages are basically inspecting the blood as it comes through and saying, are you good? You're good. Uh-oh, your uniform's not ship-shape anymore. You need to come with me. I'm breaking it down. Pretty much. That's how it goes. So the whole process of blood cell formation is erythropoiesis. It occurs in the red bone marrow. It's a myeloid stem cell, as we saw earlier. And this myeloid tissue that's in the red bone marrow is located specifically in the vertebrae, the ribs, the sternum, skull, scapula, pelvis, and then all of the proximal limb bones like our femur and our humerus. So the fatty yellow bone marrow can convert red to red bone marrow if there's like a huge amount of blood loss in the body to help increase the rate of red blood cell formation. But generally speaking, this is just occurring in the red bone marrow that's existing within those specific bones. So, in other words, you don't have red bone marrow like in your, phalanges, right? They're super small. So, how does this happen? Our developing red blood cells basically absorb the amino acids, absorb the iron that's floating around in our bloodstream. So, where do the amino acids and the iron in our bloodstream come from? The food that we eat, right? Our digestive system, anything that we put into our body. So, we're putting these nutrients into our body. Our cells are breaking them down, releasing amino acids and iron, through the blood. And then our red bone marrow is soaking that up. Our red bone marrow uses that to create hemoglobin. So the red blood cells go through this process of erythropoiesis. They start as proerythroblasts. From proerythroblasts, they're going to develop into a basophilic erythroblast. This is a cell that's basically creating and filling up with hemoglobin. A normoblast is the small little stage where the nucleus is extracted. So in other words, once the red blood cell has all the hemoglobin that it needs, it kicks the nucleus out. It doesn't need the nucleus anymore to create the protein, right? Because protein comes from RNA, RNA comes from DNA, DNA is in the nucleus. Got it. So once that all the hemoglobin's made, there's no need for a nucleus anymore. The normoblast kicks out the nucleus. It's now a reticulocyte. The reticulocyte has the majority of the red blood cell of the hemoglobin that's needed to create a red blood cell. Then it enters the bloodstream, and after it's circulating the bloodstream, it's picking up other nutrients, it's furthering its development, then it matures. So the reticulocyte is the stage of red blood cell formation where the nucleus is no longer there and it enters the blood circulation. It's no longer hiding in its little nest in the red bone marrow, it's out in the real world, aka the blood. So let's break it down. In our bone marrow, we have our proerythroblasts, which then morphs into our basophilic erythroblast. This is where lots and lots of hemoglobin is accumulating. A polychromatophilic erythroblast, which was left out of that last slide. It's basically a little sub-stage. And then the normoblast. The normoblast kicks out the nucleus after most of the hemoglobin has been synthesized, and then our reticulocyte is left, and that enters the bloodstream. So let's talk about the recycling. So in the spleen, the liver, the bone marrow, there is a lot of macrophages. Macrophages are, again, the Pac-Men of our body. We're going to be hearing about them a lot this semester. Just think of macro Pac-Man, Mac-Pac, right? Put those two ideas together. They're basically monitoring the conditions of our blood, of our circulating red blood cells, like I said, kind of up for inspection as they come through the spleen, the liver, and the bone marrow. And any old red blood cells, ones that look like, they're just falling apart, they're going to engulf them. And so the process of red blood cell engulfing happens. And this happens in order to prevent them from rupturing. Because once they rupture, then all our parts can go everywhere and we can't grab them and reuse them. So hemolysis is the process of red blood cell rupturing, and our macrophages try to prevent that from happening. So all the hemoglobin molecules, all the cell fragments that were in that make up our red blood cell are engulfed by our macrophage. And so our macrophages are basically going to store those extra parts, store the extra iron, or the iron can actually be released back into the bloodstream and then soaked back up into the plasma, and they're basically stored on a little, kind of like a little molecule boat, as I think of it. Transferrin is just this little molecule boat in our blood that holds the iron. So, overall events in our macrophages. Globular proteins, basically disassembling all of the amino acids. They take the heme protein, or that heme molecule, and they convert it into something called biliverdin. Biliverdin is converted into bilirubin. Bilirubin goes into the bloodstream. So there's this whole process of recycling the heme that takes place inside of the macrophage. Hemoglobin that's not phagocytized can break down the protein chains and is then eliminated in the urine. If there's an abnormally large number of red blood cells that have been broken down, you can have red or brown urine. It's called hemoglobinuria, where you have blood in your urine. So let's take a look at this whole process, and I strongly suggest drawing it out and recreating it on your own paper, right? On your own paper. So I'm trying to think, should I do that? I don't know. I think that would be redundant if I just redrew it. It's right here. I don't know. So the beginning process starts with the reticulocyte coming out. I suggest drawing this as I say it. How about we try that? All right, and I'll get your feedback. And if you don't like drawing it as I say it, let me know, and I will draw it as I say it, but I'll have my face, and I'll draw it on my handy-dandy little whiteboard of knowledge here, okay? So the reticulocytes go into the bloodstream. The reticulocytes do not have a nucleus. They mature about 2 days or so, and now they're a mature red blood cell. The mature red blood cell floats around for about 120 days, and then gets old, damaged, worn out, right? It's just old. It happens. It doesn't have mitochondria. It doesn't have anything in it to help it maintain its properties, right? So it gets old very quickly. And when it passes through the red bone marrow or the liver or the spleen, those macrophages that are sitting there notice that it's old and say, you know what, I'm going to eat you up before you rupture, right? And so when it eats it up, it's going to basically break down the hemoglobin, and the iron can be released back into the blood in the form and be transported by transferrin. Transferrin can take the iron back to the bone marrow or back to wherever and then put it into another hemoglobin. Or the iron can just stay in the macrophage. The macrophage can store it. The heme is broken down into biliverdin, and biliverdin is converted into bilirubin, which is then eliminated in our urine. So bilirubin can be released from the macrophages. It binds to a protein called albumin. Albumin is one of our plasma proteins. Goes out into our bile, and the bile is released in our digestive system. So any blocked bile ducts or the inability to produce bilirubin can cause increased levels of bile in our blood. This can accumulate in our peripheral tissues, right? This causes that yellowish appearance that we call jaundice, the yellowish discoloration of our eyes and of our skin. Can be created if our liver is not functioning properly. Very common in people that abuse alcohol because alcohol damages the liver. So there's also a set of steps that occur in the large intestine. So the bacteria are taking the bilirubin that's from the bile and basically converting that bilirubin into urobilins and stercobilins, and those are kind of the yellow-brown parts of our feces. So it continues to change and be modified in order for elimination. There's also some events that occur at the kidneys. So sometimes our hemoglobin is floating around in the blood, and that is going to be basically absorbed at the kidneys and eliminated at the kidneys. This can give urine its yellow color because the kidney is going to convert the hemoglobin into something called a urobilin. So the presence of intact red blood cells in our urine is usually after some type of damage in our urinary system, and that can be used as a diagnostic. We're going to have a chance to do a urinalysis later on in the semester, and you'll get to see if you have blood in your urine. Hopefully you don't. So here's the process. The bilirubin is bound to albumin. It comes into the liver from general blood circulation. The liver is going to convert it into take that bilirubin, put it into the bile, right? The bile is going to be excreted into our digestive system, eventually getting to our large intestine. And from there, it's going to be converted into urobilins and stercobilins, and it can either be absorbed in the blood and excreted at the kidneys or eliminated in the feces. So here's the whole big picture from start to finish. Let's put it all together. So we have our red blood cell formation. Again, we're just talking about red blood cells here. Proerythroblast to a basophilic erythroblast, eventually normoblast, kick out the nucleus. Reticulocyte released in the blood. Think about that, reticulocyte release, right, RR. The reticulocytes released in the blood, matures after about 2 days. Our mature red blood cell circulates for 120 days. Finally, it's engulfed by a macrophage in the spleen, the liver, the bone marrow. This macrophage can take the iron and store it, or release, does it have it here? I don't, it doesn't have it. Sorry. Or transfer it to transferrin, which takes that iron back to the blood. If I were you, I would draw a little arrow here, taking the iron back to our red bone marrow via transferrin, that little protein. So, coffee. The heme is then converted into biliverdin. Biliverdin is converted into bilirubin. Bilirubin binds to albumin, which is a plasma protein. Think of plasma proteins as like a little boat, right? So bilirubin is riding in this little albumin boat to the liver. The liver puts the bilirubin in the bile. The bile is released into our digestive tract. The bilirubin is now converted into a urobilin or a stercobilin. The urobilins can go to the kidneys and be excreted and eliminated in the urine. The stercobilins are eliminated in the feces. So hemoglobin that's not phagocytized, that's just floating around from, before the macrophage got a chance to gobble it up, can break down into its alpha and beta chains, and then eventually it's excreted. So let's think about this for a minute. You have a bruise, right? Let's think about the physiology of a bruise here, knowing now what we know about this recycling process. You get punched in the arm, okay? And a day later, you have a bruise there. What is that bruise representative of? Well, the bruise is representing your damaged blood vessels, right? When you get punched very hard, your delicate blood vessels are going to break and release blood. Usually it's the capillaries that are broken. So those capillaries are broken, and now you have this big kind of pool of blood in this area. So on the first day, you might see it look purplish, right? That purplish is the blood that's underneath your skin, right? After 3-4 days, that purple starts to fade, and what color is it? That's my dog eating something out there. It turns a yellowish green. Guess what that yellowish green is? Look at our chain of events here. That yellowish green is the biliverdin and bilirubin, right? Our macrophages, which come to that area, which are always there as our first line of defense, gobbling things up, come to that area and start breaking down all the damaged tissue, start the recycling process. So in that area, this same process will occur. So you have this buildup of green and yellow pigment, which is the biliverdin and bilirubin. And slowly, as the albumins come to take all of that bilirubin away, that yellow-green color fades, and your vessels repair themselves, which is a whole nother video that we'll make. So that is the basic breakdown and recycling of red blood cells, whether it happens naturally, like it's supposed to in these macrophages and in our liver and in our bloodstream, or if it just happens by accident or by injury, right? So that's the whole process. You can stop the video here. Actually, I'm going to stop the video here, and you can answer these 3 questions. I am a huge proponent of going through and making graphic organizers or flow charts of this stuff. Flow chart it out. Even if you take the pictures out of it, look back at that drawing on that last slide that I had, not the one with the questions on it, but the one before that. Take a look at that and chart it out. Make a sequence chart of it. Start at the beginning and just use words and arrows to plot yourself through the whole process. I guarantee it's going to help it sink in. You want to take it a step further? This is what you do. Each step of the process goes on a note card, right? So you have step 1, proerythroblast. Step 2, normoblast or whatever. Step 3, reticulocyte, right? Then you'll have all of these note cards with all these words on them. Shuffle them up. See if you can put them in order, right? That's an awesome way to study. Very interactive, very engaging, and it's active. Anything active you can do, that's the way to do it. All right, I am stopping the video here. Go back, review, answer those questions. Let me know what questions you have. I'd love to help you. See ya.